and thanks for the opportunity to be here and present the results of this very exciting collaboration. I think we've been very impressed with this data, and I hope that you will be too. So what we did was we used the Tapestri single-cell sequencing platform to examine polyclonal resistance mechanisms to FLT3 inhibitors. So I thought I'd start by providing a little bit of background in FLT3 in AML. So FLT3 is the most commonly mutated gene in AML. This has been shown in multiple large-scale genomic sequencing efforts of AML, including the TCGA sequencing of over 200 AML genomes, and a more recent sequencing effort of over 1,500 AML genomes published a couple years ago in the New England Journal. FLT3 is a receptor tyrosine kinase. It is represented in a cartoon format here. The most common mutations in FLT3 come in the form of internal tandem duplication mutations that occur in the juxtamembrane domain of the kinase. This is an autoregulatory domain for the kinase, and mutations in this area cause constitutive kinase activation that is ligand independent. And this occurs in about 20% of AML. Another 5 to 10% of AML has point mutations that occur in the activation loop of the kinase, largely at this residue G835. And these are also constitutively activating. It's been known for a long time, actually, that patients with ITD mutations actually have decreased overall survival and decreased progression-free survival. And this is largely due to increased relapse rate for this particular population of patients. So given this known propensity for poor prognosis associated with FLT3 mutations and the fact that it's the most commonly mutated gene in AML, many efforts over the years have focused on developing clinically active inhibitors of the FLT3 kinase, and a number of these have come into clinical development over the years. Unfortunately, they have all been limited by the development of resistance, and this has largely been due to on-target mutations in the tyrosine kinase domain. So this shows a variety of FLT3 inhibitors that have come into clinical development over the years that have shown some activity as monotherapy, and all of them have been associated with resistance due to on-target secondary mutations within the FLT3 kinase domain that impair drug binding. And you'll notice that the most common of these are actually secondary mutations that occur at that activation loop residue D835. And so one of the things that we were interested in understanding is the degree of heterogeneity that can occur for patients that develop resistance on FLT3 inhibitors. So we undertook a single-cell sequencing project a number of years ago, actually, where we just examined the FLT3 loci at the ITD locus as well as at the D835 locus. So this represents the results of this effort in which we sequenced about 300 cells from each of these patients at the time of relapse. And each of these patients is represented here in these squares or rectangles. And we represent the genotype of the population of cells with these circles. So the ITD zygosity is represented here on the right, in which one red square represents heterozygosity for FLT3 ITD, and two red squares represents homozygosity. On the left here, we have the D835 mutation zygosity. Again, one colored quadrant represents heterozygosity and two colored quadrants represents homozygosity. Each individual color represents a different D835 mutation that was found. And then finally, the relative size of the circle represents the relative proportion of the cell population that had that particular genotype. And so what we were able to show is that in all these patients, there was a very polyclonal mix of heterozygous and homozygous FLT3 ITD and D835 mutations that seem to be associated with clinical resistance in these patients. As you can see, for example, in this particular patient, there were actually 3 different mutations at the D835 loci, all associated with either heterozygous or homozygous FLT3 ITD mutations. And then in 2 of the 3 patients, we actually were able to demonstrate that there was a proportion of cells that had only D835 mutations alone. Now, this mutation alone is actually known to confer resistance to the FLT3 inhibitor in question, which was quizartinib. But it was a surprise to identify a clear population of cells that appeared to not actually have a ITD mutation co-occurring with the D835 mutation, suggesting that there's actually a degree of polyclonality in terms of the FLT3 genotype in each of these patients. And the real surprise to us was actually in all these patients, we identified a significant proportion of the cellular population, 30 to 50%, that seemed to have an oncogenic FLT3 ITD mutation, but no other D835 mutation to explain the resistance that was seen in these patients, suggesting the co-occurrence of off-target resistance in all of these patients. And then we also found in a smaller number of cells that there were some cells that had neither a FLT3 ITD or D835 mutation, which either suggests the presence of contaminating normal cells in these cellular populations or a non-FLT3 driven blast population. So we were able to demonstrate using a very actually kind of low throughput fashion with PCR amplification that there was a surprising degree of polyclonality in these AML populations, even if you're just looking at the FLT3 loci. So given that we know that D835 is such a major clinical problem, a lot of effort over the years has actually been put into developing inhibitors that have activity against this particular mutation. And the best in class of these is a drug called ASP2215, also known as gilteritinib, which is a type 1 inhibitor of AXL and FLT3. It's highly potent against FLT3. And it actually has very good activity against that drug-resistant D835 mutation, both in vitro and in vivo studies. A phase 1/2 clinical trial of this drug in a FLT3 mutant AML in patients who were relapsed refractory to initial chemotherapy showed that ASP2215 is actually highly active. It achieved approximately a 40 to 50% composite complete remission rate in this highly refractory population. As is shown here, this responses were achieved at dose levels starting from 20 milligrams on up to the maximally tolerated dose of 300 milligrams. But unfortunately, as you can see by this survival curve, despite the initial responses, most patients still relapsed and went on to die of their disease. So we undertook an effort to understand the mechanisms of relapse to this particular drug, and we looked at patients who were treated at the clinically active dose of greater than 80 milligrams a day. And we analyzed serial samples from these patients to see what, if any, mutations were acquired at the time of relapse. And sequencing of 25 subjects identified new mutations at the time of progression in about half of subjects using a targeted next-generation sequencing panel. And interestingly, only a small subset of the mutations identified were within FLT3 itself, suggesting that most of the resistance mechanisms to this next generation FLT3 inhibitor were actually going to be more off target. And then most impressively, half of the mutations that were identified were actually activating oncogenic RAS mutations, either in NRAS or KRAS, suggesting that oncogenic RAS was going to be a major mechanism of resistance to this particular drug. And when we looked closer, we noted 2 patterns of occurrence of these RAS mutations. In some subjects, what we saw was that the RAS mutation was undetectable at the time of study start, and then clearly was acquired at the time of drug resistance, whereas we saw maintenance of the FLT3 mutation variant allele fraction at that's similar from study start to study end, suggesting there might have been a small population of double mutant NRAS and FLT3 mutant cells at the time of study start, or that this double mutant population was somehow acquired and enriched throughout the course of the study. In a second pattern of cells, of resistance, we noted that there were some patients who actually lost their oncogenic FLT3 mutation, so they lost the FLT3 ITD mutation, and instead had emergence of an NRAS mutation that perhaps represented a non-FLT3 driven AML blast population that was selected for on the drug. So we noted that, therefore, there could actually be two different mechanisms of quote-unquote off-target resistance to this drug, either reactivation, a bypass mechanism in which you see reactivation of downstream MAP kinase signaling mediated by oncogenic RAS, or actually selection for a non-FLT3 driven RAS-driven clone. And we knew that in the laboratory, when we put oncogenic RAS into FLT3 ITD expressing cells, that it is able to confer resistance to the FLT3 inhibitor, as is shown here in this proliferation assay. Cells that have FLT3 ITD, which also express oncogenic RAS mutations, are able to proliferate in the presence of gilteritinib. And they also demonstrate resistance to apoptosis as measured by induction of caspase-3. And you can see the live cell, percentage of live cells in the oncogenic RAS-containing cell lines compared to the parental cell line. And this is also associated with maintenance of ERK phosphorylation, as you might expect in the RAS mutant cell lines compared to the parental. However, we really wanted to understand the true clonal heterogeneity and clonal selection that was occurring in these patients, and how much of the resistance was actually due to co-occurrence of oncogenic FLT3 and RAS mutations versus selection for a non-FLT3 driven clone. And to do this, we utilized the Tapestri platform. And so this represents a patient that was analyzed at the time of resistance using the Tapestri platform. And this is a patient who was on drug for a number of months. And this is her resistance sample. And you can see here that if you compare the variant allele frequencies that are obtained from bulk sequencing, so this is actually bulk, just regular next generation sequencing of the bulk population, compared to the variant allele frequencies that are extrapolated from the Tapestri single cell sequencing, they're actually very similar, as you can see here. There are some loci that are not captured using the Tapestri platform, either due to the fact that the panel does not contain that particular genetic loci, or due to technical aspects of the sequencing or sequencing analysis. Here we show hierarchical clustering of over 4,000 cells obtained in this relapse sample, where each of the lines is a particular cell, and each of these bars represents a mutation. The dark red represents homozygosity, and the light red represents heterozygosity. And what you can appreciate, I think, from this is that this pretty much the vast majority of this relapse sample was heterozygous for DNMT3A, heterozygous for NRAS, and homozygous for FLT3 ITD and WT1. And this is broken down here where we show the clonotypes. So this, the vast majority of the resistance in this patient is actually due to a cellular population that appears to be driven by oncogenic RAS, but is quadruple mutant for FLT3, WT1, and DNMT3A. You'll also note the appearance of a small population of cells that appears to be mutant only for FLT3, NRAS, and WT1. And if you know anything about AML, you'll think about AML biology, you'll think that this is unusual because typically DNMT3A mutations are actually thought to be ancestral mutations and actually occur in the pre-leukemic stem cell. And so what we think is that this represents another potentially technical artifact of the pipeline of the sequence of the platform, in which you can see allele dropout, and that of the mutant allele, and you can see that when populations are approximately 8 to 10% or less, it's unclear if they may likely be due to allele dropout or potentially represent a real cellular population. And further investigation of serial samples can usually help elucidate this, and we'll show you an example of that later on. This is another resistance sample from one of these patients, and again, you'll note that largely This sample appears to have co-occurrence of the FLT3 mutation, NRAS, and WT1 mutations that represents the resistance population, and a smaller population of cells that is likely represents the ancestral clone that is only double mutant for FLT3 and WT1. And finally, a more complex case. So again, this is another case that appears to have, actually, in contrast to the other two, multiple genotypes and clonotypes represented in the resistant sample. You'll first note that, again, the variant allele frequencies obtained from bulk sequencing compared to the variant allele frequencies obtained from single-cell sequencing are very similar. And then you'll notice that at the time of relapse, there appear to be two dominant populations. One is not the expected NRAS FLT3 mutant population that also appears to have an ancestral IDH2 SF3B1 mutation in the background. But there's also a very large population of cells that is mutant only for IDH2 and SF3B1, which represents a non-FLT3 driven population that potentially was selected for during the course of drug treatment. And then again, you'll note smaller populations that may or may not be due to mutant allele drop, that may be technical artifacts due to mutant allele dropout, although it's likely either the SF3B1 or IDH2 population potentially were also ancestral clones of this IDH2 SF3B1 population. But to further elucidate this and understand it better, we conducted sequencing of serial samples from this patient. And what we were able to demonstrate, looking at a pre-treatment sample here, a sample that was obtained 28 days after the start of drug treatment, and then a sample that was obtained a couple months after that, and finally a sample that was obtained at the time of relapse 6 months later. So what we'll notice is that IDH2 SF3B1 ancestral non-FLT3 driven clone was clearly selected for during the course of drug treatment, suggesting that yes, indeed, this is drug selection for a non-FLT3 driven cell population on a drug treatment. And conversely, you can see that we start off with the very large FLT3 mutant IDH2 SF3B1 drug-sensitive population that also slowly goes away on drug treatment. And then finally, most excitingly, we see that actually the relapse clone, the second relapse clone that is driven by NRAS and has the FLT3 NRAS IDH2 SF3B1 genotype, actually can be detected quite early on the time of drug treatment. So even after only 28 days on drug, we can already detect a small population, 0.1% of cells, that actually has this drug-resistant NRAS mutation. And this is clearly selected for on a drug treatment. After about 4 months on treatment, you can see that there is about 1.3% of the cellular population that's represented by this NRAS mutant clone. And then finally, the relapse clone at the time of frank relapse represents about 26% of the cellular population. And what this slide shows you is how nicely the clonal architecture is represented by the single cell sequencing compared to what you cannot resolve based on just looking at bulk sequencing alone. So what this represents is the variant allele frequencies that would have been obtained from just bulk sequencing of this population. And you can see two very clear things. One is that you cannot resolve that there is a cellular population that is clearly not FLT3 dependent, this SF3B1 IDH2 population, which basically is represented by stable variant allele frequencies throughout the course of treatment. But when you look at the single cell sequencing, you can see clear selection for this relapse clone that is non-FLT3 driven. And the second thing, and very important, I think, for use in a clinical scenario, is that the NRAS mutation cannot be detected until the time of frank relapse when you look only at the bulk sequencing. However, when you look at the single cell sequencing, you can clearly detect very early on treatment the emergence of a drug resistance NRAS mutant clone. So potentially this has great power when you think about the fact that in a clinical scenario, if you knew this, you may want to add another treatment or even switch treatment to avoid this relapse scenario. And this is shown very nicely here in this fish plot where we actually see evolution of the cellular populations during the course of drug treatment over time. So as you can see here, we start out with a very large IDH2, FLT3, SF3P1 drug-sensitive population that goes away on drug treatment. There's a smaller population of cells that probably represents the ancestral clone that is non-FLT3 driven, that's also clearly selected for on the course of drug treatment. And then finally, here is a secondary drug-resistant population with an NRAS mutation that is not, that you cannot see pre-treatment, but 28 days, only 28 days after starting drug treatment, you can already see emerge, and it's clearly selected for on the course of drug treatment. And this is data that uniquely was obtained only because of the single-cell sequencing. So in summary, we know that clinical resistance to FLT3 inhibitors is clearly polyclonal. It's due to on-target tyrosine kinase domain mutations, which now with newer and better FLT3 inhibitors may be less of a clinical problem. And then now emergence of off-target resistance, either through to reactivation of downstream signaling, maybe largely due to oncogenic RAS mutations. And then secondarily, selection for non-FLT3 driven clones. And so the power of the single-cell sequencing was able to demonstrate in patients both clonal selection for an oncogenic RAS-driven clone, as well as for pre-existing non-FLT3-driven clones that are selected for in treatment. And with that, I'd like to thank everybody that was involved in the project, particularly Mission Bio, and I'm happy to take any questions.
Just a simple question. How did you get from your sample to those individual cells? What were the sample pre-processing steps?
So the question was, what were the sample pre-processing steps that were necessary? And actually there were none. These were just frozen, viably frozen peripheral blood mononuclear cells or bone marrow mononuclear cells that were thawed in a standard manner and then subjected to the sequencing. So about 100,000 cell input. So we actually, a lot of these, we very typically freeze down millions of cells from these patients at the time of relapse. And so cell input was obviously not an issue.